Let me start by thanking the Executive Committee of eScan for giving me the opportunity to talk to you today. I'm really sorry that I couldn't be there in person, but maybe this is the next best thing. I'm going to deliver the talk that I would have given you if I were there, and you should feel free to follow up with me by email if you have any questions. What I'm going to talk to you today is really about the nature of emotion, and I'm going to use the science of emotion to try to pave the way for a new approach or an emerging approach to understand how the brain and the body create a human mind in the context of other brains and bodies, which are also creating minds. And the starting place for us is something that I like to call the classical view of emotion. This is the dominant view in the science of emotion. For those of you who know something about emotion theory, this encompasses the basic emotion approach and also causal appraisal approaches. The key set of assumptions that unite these theoretical views is that there are essential emotion categories, right? So the idea here is that each category of emotion has an essence. That essence may be a physical fingerprint, like an autonomic nervous system signature, or a specific diagnostic facial expression, or a particular set of neurons in a region of the brain, in a network of the brain, in a pattern in the brain, a brain state. But the idea is that all the instances of a category are similar in some way, and that similarity has some physical basis in reality, which we would refer to as the essence. And so what I'm going to do today is I'm going to suggest to you that this is actually not, this assumption of essentialism is not really supported by the scientific evidence. I'm going to give you 3 examples of essentialism in the science of emotion, and then I'm going to use that as a platform to discuss what in my lab and in others around the world see as an opportunity for a paradigm shift. Now, I should say that if you look through the history of science, every science begins with essential categories, the assumption that instances that are similar within a category have some physical basis in nature. And then there's this slow shift away from an essentialist view to a more context-dependent, holistic view. This is true in physics, it's been true in chemistry, it's true in biology, away from essentialism towards a more holistic contextual science. And so in the next 40 minutes or so, I'm going to do this, sort of pave this path for us today. Give you 3 examples from the science of emotion, of essentialism, and then outline the emerging paradigm shift that's happening right now, at least as I see it. So the first assumption, essentialist assumption in the classical view, is that emotions are displayed on the face with expressions that we recognize. So people are supposed to smile when they're happy, they're supposed to frown when they're sad, scowl when they're angry, and so on. Everyone around the world is supposed to be able to recognize smiles and frowns and scowls as expressions of emotion. But consider this evidence. Here are the facial configurations, which are presumed to be universal expressions of each emotion category. How frequently are these facial configurations actually made during episodes of emotion? And the answer is not so much. The white bars here represent meta-analytic averages, average proportions, of the times that people make these facial configurations during actual emotional events. The blue bars represent the correlations, the average correlation between the stipulated expression and some other measure of emotion, like a self-report or an autonomic measurement. And so what you can see is that in real life, people move their faces in a variety of ways, and in a given emotion, there is only weak evidence that a specific face is made consistently to express that emotional category. Now, I want to make it really clear here that almost all of these are above chance, right? So we're not saying that facial movements are random here, but we are saying that, for example, about 30% of the time, give or take, people make a wide-eyed, gasping face in fear. And it's also notable that across cultures, that particular face is often perceived as something other than fear. So for example, in Melanesian society, it is the symbol of a threat anger face. So in real life, people move their faces in a variety of ways to express a given emotion. People usually see this person as expressing grief or pain or sadness or fatigue. This is actually my daughter, Sophia, from a couple of years ago at the Cologne Museum of Chocolate, expressing only what I could describe as a profound sense of pleasure. And this little sweetie is also expressing a profound sense of pleasure. And so the lesson here is that people move their faces in many different ways during instances of the same emotion category. Now, if we look at this little guy's, just look at his, the facial movements around his eyes, we can see that he is making a facial movement that is very similar to the presumed universal expression for anger, a scowl. And in fact, people often see this face as anger. But in fact, this is Jim Webb, who won the US Senator's race in Virginia and returned the Senate, the US Senate, to Democratic control with his victory. Without context, we see his face as communicating anger because, in fact, this face symbolizes anger in our culture. So people not only move their faces in different ways during the same emotion category, they also move their faces in the same way during different instances of emotion, of different emotion categories. What about the idea that each emotion has its own autonomic nervous system signature or fingerprint? Well, here's another meta-analysis, this time from our lab. This is 233 papers of non-clinical subjects. The effect size is on the x-axis, and the type of autonomic measure is on the y-axis. Here are the effect sizes for neutral inductions. The shape represents the average effect size. Larger shapes mean we had more studies available to analyze. If the classical view is correct, then what you'd expect to see is little overlap in the means or the confidence intervals around those means, those average effect sizes, which are represented by the bars. So here are the results for anger, fear, disgust, sad, and happy. What we see is not distinctiveness. Instead, there are no autonomic features that are actually unique to any emotion category. There's tremendous overlap in the distribution of autonomic features for, across the various categories. And from this, it's really hard to imagine that there is any specific pattern that is diagnostic of each emotion. And in fact, if you do pattern classification analyses on these data, what you find is exactly that. Now, here's what you see when you look at individual studies that use pattern classification. In a given study, a pattern classification solution will distinguish people who are angry from people who are sad from people who are afraid, sometimes. Sometimes it happens. Sometimes you'll see this evidence in a given study. And in another study, you'll see, again, that a pattern classifier distinguishes people in these different states. But the two pattern classifiers don't replicate each other. They don't look the same, and they don't work on the alternative data sets, right? It's very specific to that particular study. And this is often seen as a failure to replicate, but in fact, I don't think that this is a bug. I think this is a feature of the phenomenon itself. And it's a feature that we're going to revisit in a second now when we talk about the assumption that there are dedicated emotion neurons in the brain, that each category of emotion can be localized to a particular brain region, a particular circuit, a network, or maybe a distributed pattern across the brain. So what I'm showing you here are the pattern classifiers for brain imaging studies of emotion over the past 20 years or so. The first thing to notice here is that the emotion patterns are distributed across the brain, so they're whole brain events. They are not localized to individual regions or networks. People often mistake these patterns for the neural essences for emotion, that is the brain states for anger, disgust, fear, and so on. But that's actually incorrect. These are not biomarkers. These are not fingerprints. These are not signatures, right? These are statistical abstractions, meaning different instances of sadness will be associated with different distributed patterns of activity across the brain. To be correctly diagnosed as an instance of sadness, the pattern of activity just has to be closer to the blue pattern than to any of the other patterns. A given instance can be correctly identified even if it shares no features with that blue pattern, meaning there's not a single voxel of increased activity that is common between the pattern and the correctly identified instance. And to put this into context, it's sort of like saying, you know, because in the US, the average middle-class family has 3.13 people in it, but we don't expect every family to have 3.13 people, and in fact, as far as I know, no family has 3.13 people in it. So classification patterns are not brain states. They are not fingerprints or biomarkers. As I said, they are abstractions, statistical abstractions. And in fact, when you look at pattern classification analyses across studies, they don't replicate. When you look at them, even across different individuals, they often don't replicate. So a category of emotion like sadness is a highly variable population of instances. And now for those of you who think, well, you know, brain imaging technology is just too coarse for us to find any specificity and any, you know, to identify the neural essences of emotion, I'm showing you here evidence from 2 monozygotic twins who have amygdala lesions. What you're looking at here is an axial view of a structural scan with the, you know, I'm talking to a neuroscience audience who understands, but I'll just say with the top of the brain, with the front of the brain at the top of the image. These 2 sisters are monozygotic twins. They both had calcified amygdalas at around the age of 12. They have a fully lesioned amygdalas. They grew up in similar circumstances, have similar IQ, live down the street from each other now as women, as married women. They're basically similar in their life experiences, but Sister BG has fear-related deficits. That is, she can only experience fear under extreme circumstances, like a carbon dioxide load, whereas Sister AM has no fear-related deficits whatsoever. This is an example of degeneracy. In biology, the concept of degeneracy is meant to convey that a given outcome, here we're talking about the experience or perception of fear, a given outcome can be caused by more than one mechanism or more than one structural correlate. And so these data, along with others, lead us to understand that sometimes the amygdala is important to the perception of fear, sometimes the amygdala is important to the experience of fear, but it often isn't. There have to be degenerate ways that a brain can create the perception of fear or the experience of fear. So this basic summary here is that variation is the norm when it comes to emotion. And when I say that, I'm not saying that everything is random and there's nothing reliable, nothing predictable, nothing meaningfully patterned. I'm saying that what the data look like, these are, and you know, consider that most of the studies that I'm referring to are summarizing data from experiments that are designed to model the clearest case, right, the prototype, as it were, of an emotion category, and yet we see tremendous variability. So the suggestion here is that that your brain is able to create a population of angers, a population of sadnesses, a population of fears, and so on, each one tailored to the specifics of the environment. Now, at some point, Some of you might be thinking, well, no one really believes that every instance of an emotion category is exactly the same as every other instance. That's just preposterous. And you're right, that's true. At issue is how much variation is there within a category, how similar are the instances across categories in their physical features, and how intrinsic is this variation to the nature of emotion. So what I'm showing you here is a theoretical space that we can use to depict the degree of essentialism in various theories. The x-axis refers to the similarity in surface features, like facial expressions or autonomic changes. And along the y-axis is there a deep similarity, like a neural similarity, between all the instances of an emotion category? Or is there some hypothetical mechanism that's unknown, like an affect program? That would be an example of what cognitive scientists refer to as psychological essentialism. And so here are I've just picked a selection of theoretical views to display them for you. The red zone here represents categories that are thought to be Aristotelian or classical in their structure. The idea is that the instances are all similar to each other in a physical sense, both in their surface features and in their deep features. The yellow zone is meant to depict scientific approaches that assume some variability. So an emotion category is thought to be a prototype category, where there's a single prototype, which is either the most common or the most typical instance of the category, but then, there's variation in the instances around that single representation. And the green zone is thought to represent theories, approaches that propose emotion categories as abstract, ad hoc, conceptual categories, meaning that a brain is creating a category, an emotion category, on the fly in a way that's tailored to the environment in order to best meet the goals of that emotion in that situation. And that's what I'm going to talk to you about for the rest of today. So I'm going to talk to you about the work that we do in my lab, which is guided by the theory of constructed emotion, which assumes that emotion categories are abstract conceptual categories that the brain creates on the fly as they're needed. Now, normally in the science of emotion, actually in the science of psychology more generally, since psychology became a science, scientists start with a folk psychology category that's been bequeathed to us from ancient Greece. In our case, it's anger and sadness and fear and surprise and happiness and so on. And then they go looking for the physical basis of that category in the body, in the brain, in the face. This has never been a particularly fruitful approach, not really since, people have been trying to find the physical basis of categories since the late 1800s and really haven't been able to do it. So in my lab, we take a really different approach. We start with the brain and the nervous system more generally. So we start with the brain and the body, and we ask the question, well, given the kind of brain that we have, that's being carried around by the kind of body that we have, what would be the kind of emotions that we would make? So we start with the biology, and then we ask questions about what emotion might look like in that kind of an organism. And so we start right off the bat by looking at the brain's fundamental problem from the brain's perspective, which is that it's constantly faced with a reverse inference problem. So if you think about it, your brain for its entire life has been entombed in a dark, silent box called your skull. It has to learn what's going on in the outside world only through scraps of information that it receives from the body's sensors, so that it knows what to do to keep you alive and well. so that you do your most important task, which is to propagate your genes to the next generation. So really what it's trying to do is find the causes of sensations when all it has are the effects. So it receives information from the world, but any given change in ear pressure, any given flash of light, and so on, can have multiple causes. And in addition, that information is not always, doesn't always really have high fidelity. Sometimes it's incomplete, sometimes it's noisy, and so on. And it turns out that your brain has the same kind of reverse inference problem with respect to sensations from your body. So it receives sensory inputs from the body, which we call interoceptive inputs. But these inputs are noisy. They are often incomplete. And any given sensation can have multiple causes. So an ache in your gut could be hunger if you are sitting at a dinner table. That exact same ache in your gut could be anxiety if you're in a doctor's office waiting for a test result. And that gut feeling might also be evidence that a defendant is guilty if you're a judge sitting in a courtroom, particularly if it's right before lunch. So your brain is having to constantly guess at the causes of sensations when all it has are the effects. How does it solve this fundamental problem? The answer is it has one other piece of information available to it, one other source of information available to it, and that is past experiences. So your brain is remembering past experiences that are similar in some way to the present sensory array in order to make sense of those sensations. So I'm not what I'm suggesting is that the brain is not asking, what is this? What is this sound? What is this sight? What is this ache? It's asking, what is this like based on my past experience when changes in the world and in the body were similar to the present conditions? In psychology, a group of things that are similar is called a category. And a representation of that category is called a concept. So here's the hypothesis. Your brain is using past experiences to create concepts on the fly as ad hoc concepts. So it's reinstating a variety of patterns from past experiences, either in total or combining them in a new way, so that it's creating representations that are similar to the present conditions as a way of making sense of the current sensory conditions so that your brain knows what to do next. And the cool thing is that evidence from anatomy and from physiology and from functional studies indicate to us that the brain is doing this predictively. These ad hoc concepts are your brain's best guess about the meaning of upcoming sensory inputs and the relevant motor changes that are necessary. So you can think about these ad hoc concepts as possible causal relationships between events in the world and in the body as they are right now, right this moment, and their sensory and motor consequences in a moment from now. And I'm going to suggest to you that this is the default process by which the brain navigates the world and constructs your experiences and guides your actions. So it goes something like this. Your brain starts with initial conditions in the body and in the world and projects forward in time about what the body has to do next. Motor actions, the visceromotor changes in the systems inside the body to support those actions, and so on. Efferent copies of these motor and visceromotor commands become the predicted sensory consequences of those actions that actually change the firing of sensory neurons. So those efferent copies are, the equivalent of what did the brain see last time the body was in this state preparing for this action? What did the brain hear the last time the body was in this state preparing for this action, and so on and so forth? These predictions, these concepts, are candidates for explaining causally the incoming sensory inputs. And then inputs from the body and the world have the opportunity to either confirm those predictions, that is, the incoming sensory inputs match the firing in sensory cortices, in primary sensory cortices, those cortices are firing, in a pattern that are the predictions, basically, of the upcoming sensations. If the sensations from the world, the sensory information from the world confirms those predictions, then we say that the sensations have been categorized, making them meaningful so that the brain knows what to do about the next. But inputs from the body and the world can also modify predictions. That is, if there's some difference between what the brain expects and what it receives, that difference is called prediction error. And the encoding of prediction error has a fancy name. In psychology, we call it learning. So the brain learns new information that it wasn't able to predict this time around, so that it can predict better next time around. So in this view, your perceptions and actions are the remembered present, which is a phrase that comes from the neuroscientist Gerald Edelman. A remembered present that is confirmed or modified by the sensory periphery. And this is how changes in air pressure become sound, changes in light become vision, chemicals in the world become smells, and so on, and how the aches and shivers and other sensations in your body become affective feelings. Now, this is called more generally predictive coding or active inference or the Bayesian brain. There are lots of names for this approach. In fact, how you know something is important in psychology is that people come up with, they rediscover it again and again and again and give it a lot of different names. Now, at this juncture, I could, if I were giving a lecture, for example, we could just systematically go over all the evidence in support of predictive coding and ask, is there any evidence that doesn't support it? We could talk about the anatomical evidence, the signal processing evidence from the brain, the physiological evidence from the brain and the body. We could look at all the brain imaging studies that support this view. But instead, what I'd like to do, well, let me just say this, that for any of you who are skeptical that this is really how the brain works, that how your brain works, think of the last time you had a song going through your head that you could not get out of your head, that you actually hear in your head. That's a prediction. That is, or we also call that simulation, or we could call it perceptual inference. But again, all of those words mean the same thing. They mean that your brain is doing something remarkable. It's changing the firing of its own neurons in sensory cortex. In this case, it would be auditory cortex, without any sensory input from the world whatsoever. So instead of going over all the evidence from anatomy and metabolism and physiological efficiency and so on, instead of looking at all the evidence that every exteroceptive system seems to work by prediction, instead what I want to do is talk to you about where predictions originate in the brain. So I want to show you a hypothesis about the brain's predictive architecture and how the brain basically goes about making mental events and controlling action with predictions. Now, there's over 30 years of track tracing studies in macaques and other mammals that show us that there are particular parts of the cortex that are the most powerful predictors in the brain. And for the sake of time today, I'm not going to talk about all the other important predictive structures in the brain, like the hippocampus or the cerebellum, right? I'm only going to focus on the cortex because, we only have, otherwise we'd be here for many days. What I've done here is I've taken the Brodmann areas which are the parcellations of the cortex, and I've colored them in different shades of gray, following a paper by Helen Barbas that was published in 2015. And in the bottom, what you see is a depiction of the insula, which you would see if we were to take the temporal lobe and the frontal lobe and pry apart the Sylvian fissure, we would expose the insula. So it turns out that all of this track tracing work suggests that the regions where prediction signals originate, prediction signals, another name for them is also feedback signals, confusingly, or a forward model. Different literatures calling them different things. The regions of the brain that originate predictions are in dark gray and some of the medium gray. These brain regions have another name. They go by the term limbic. So here's the irony, which I think is, it's one of the great ironies in science. It's one of those moments when you just, love being a scientist because it turns your beliefs right on their head. For centuries, scientists have considered limbic regions to be the home of emotions, that they're supposed to be the most reactive parts of your brain. You're the home of your inner beast that is highly reactive and in need of control from your very prodigious prefrontal cortex. But actually, we know that these regions are the source of prediction signals that are driving perception and action in the brain, not just during emotion, events that are emotional in nature, but from the moment that you are born until the moment that you die, in every moment of your life. Your brain is predicting sensory inputs from the world and anticipating how to act on them with limbic cortices. So, limbic regions are the most powerful predicting regions in the brain. And here what I'm showing you is a depiction of the upper layers of the cortex. This is a depiction that comes from work by Barb Finley. In fact, this is her figure. And what you can see is that there's a gradient along this cortical sheet. So when we start at the back of the brain, say at primary visual cortex, and we move to the front of the brain, let's say to one of these limbic cortical regions, what we see is that we move from primary visual cortex, where there are many cells with very few connections, and the pyramidal cells are very small. And as we go to the front of the brain towards limbic regions, we see that the cells become less numerous, they become larger, and they have many more connections. And it's the gradient moves in the other direction. When we go from the front of the brain, a limbic region, and propagate back to, say, V1, the cells are getting smaller, they're having fewer connections, and there are more of them. So what does this mean computationally? Well, what it means is that when we go from a limbic region out to V1, that efferent copy, which is like what is the prediction of what I saw the last time my body was in this particular state, what's happening is that there are multimodal summaries of that limbic, the representations in the limbic regions are multimodal summaries that cascade to sensory and motor regions, becoming progressively more particularized as simulations or prediction signals. This is how a single summary of firing can influence the firing in all of the sensory and motor cortices as creating a prediction signal with very specific sensory predictions. And so you can think of this as concept generation. This is the process of ad hoc concept generation. And similarly, when we move in the other direction from V1 to the front of the brain, what's happening is there's integration across the cortical sheet. And this is happening not just in the visual system, but actually in all the sensory systems. So there's integration across the sensory domains and progressive dimensionality reduction until we get to the front of the brain, where there's a sort of a summary. And so we can think of this as concept learning. The next question that I want to address is, well, where are these limbic cortical regions? What brain networks do they appear in? Because right now, there's a tendency in neuroscience to try to think about brain function in terms of networks, the function of networks. And so using a data set of over 700 subjects, we used what are called resting state scans, which I know you know means that subjects are lying still in the scanner and they are not being probed with an external stimulus. And what we did is we asked the question, which of the regions of the brain, which voxels in the brain are correlated, which of the regions of the brain have a temporal fluctuation in their bold signal that correlates with each of those limbic cortices that I showed you before. And so we ran that study, we replicated it, and what you're looking at now, the red blobs, are actually depictions of connectivity, not activity. And we identified two networks that contain all of the limbic cortices in the brain, and those two networks overlap in cortical hubs. What's interesting here is that these are very well-known networks, the default mode network and the salience network. Both have been implicated in hundreds of experiments at this point across a range of different tasks. Of course, they go by many names. In fact, the default mode network, I think, has more names than Sherlock Holmes, more aliases than Sherlock Holmes, because scientists tend to name a network in line with the phenomenon that they're most interested in. And so today, I'm not dedicated to these labels, I'm just using them because they're the most common ones. And of course, the hubs, some of these hubs actually are called Rich Club hubs, which means that they are some of the most connected regions in the brain. And they are responsible for coordinating integration of information across networks, actually. And I want to show you that this system contains the posterior insula, which is primary interoceptive cortex. So this is the primary sensory region for all the sensations from your body. And most of the time, we don't feel those sensations specifically in any high-dimensional detail, right? We don't feel every ache, every pound of our heart, every constriction of our lungs, every gurgle in our stomach, and so on. Because if we did, frankly, we'd never pay attention to anything outside in the world ever again. Instead, we tend to experience these sensations as affective feelings, as simple feelings of feeling pleasant, unpleasant, and so on. High in arousal, low in arousal. Which is one of the reasons that we've argued that affective properties like valence and arousal are not specific to emotion, but they are actually properties of consciousness. Because your brain is always regulating your body, your brain is always anticipating and making sense of sensations from your body, and therefore your brain is always constructing affect. So this structure, which is, by the way, I should point out, these connections have been verified in track tracing studies in macaques. So not just imaging data, but also anatomical data, suggests that whatever else these networks are doing psychologically speaking, they are also representing the sensory changes in the internal milieu of your body, even when you're unaware of it. And of course, there's legions of studies which show how affect influences the perceptions and actions in everyday life. So what else are these networks doing? Well, these networks have far more extensive subcortical projections than anything that people typically discuss. And so what you can see here is I've projected the connectivity into the volumes. And so you can see the connections of the default mode regions, the salience regions, and the hubs where they overlap, which tells us that these limbic cortices are actually modulating the autonomic nervous system, the immune system, the endocrine system and all of the internal systems of the body via their connections to these brainstem regions, like the periaqueductal gray, the parabrachial nucleus, all the way down. I should have also said the hypothalamus. And so the bold hypothesis here is that whatever else these networks are doing psychologically speaking, they are also regulating your body's internal systems and representing the interoceptive consequences as affect. Now, in neuroscience, there's been a tendency to look for one-to-one correspondences between neural activity and a particular psychological function as validity evidence for what that network is doing. And when I originally started to talk about this idea more than a decade ago, I got a lot of flack from people saying, well, you can't claim that this is the only function of these networks. That's an error of reverse inference. And my answer then, as is now, and I think a decade of research has borne out my suggestion, that these networks, you can think of them as multi-purpose, in the sense that they contribute to multiple psychological phenomena while they are simultaneously regulating your body. The inability to perform reverse inference as a scientist on these data is not a bug, it's a feature of how the brain controls the rest of the nervous system and the body. And so when I say that, whatever these networks are doing psychologically, they're also regulating your body, what does that mean? What are they actually doing, psychologically speaking? And the answer is pretty much everything. So here's just one example from one slide from the default mode network showing you all the range of psychological phenomena that it's been involved in. And I guess I'll just point out that, trying to find a one-to-one correspondence between the default mode network and a particular psychological function is in itself an example of essentialism, right? As opposed to a much more situated, flexible approach to understanding neural activity. What about the salience network? Well, I could have shown you a slide that looked very much like the one that we just saw for the default mode network, but in fact, what I decided to do instead was show you a meta-analysis that my lab performed a couple of years ago. What we did is we just took the neurosynth database, so at that point, almost 6,000 studies of brain imaging studies of every domain, across every domain of psychology. We just dumped it into one meta-analysis and asked, where are the hot spots in the brain? Like, which regions of the brain are constantly showing an increase in activity across all domains? And the answer is pretty much the salience network. So to scientists who study physiology and signal processing, these findings that I'm showing you are not particularly surprising. To pull a quote from a terrific book by Sterling and Lawlin, the core task of all brains is to regulate an organism's internal milieu by anticipating the needs of the body and preparing to satisfy those needs before they arise. So this suggests that, that this predictive coding approach of understanding brain function presents a sort of a unified computational framework that places metabolism and energy regulation, which predictively is referred to as allostasis. And I should point out, allostasis is not the same thing as homeostasis. Homeostasis is, there are many differences, but for our purposes today, what we're concerned about is the predictive component of energy regulation. So whatever else the mind is doing, whatever else the brain is doing, it's also regulating metabolism and energy in an efficient way and representing sensory consequences of those allostatic changes. And this is at the core of all mental activity, not just in emotion, but also in thinking and perceiving and acting and deciding and so on. And so from a biological standpoint, it doesn't really make sense to divide emotion and cognition into separate faculties where one regulates the other or insists that they cooperate in order to control behavior. The brain is constructing predictions in the service of allostasis And all predictions, including ones that you experience as subjectively cold, contain information about interoception and have some kind of affective tone, even if they're very subtle. The implication of this is that we should be considering metabolism and energy regulation when we study any psychological phenomenon, not just emotion or stress. So if you think about it, psychology is forced because of funding considerations to run studies with reasonably small samples. And so if metabolism and energy regulation is at the core of what your brain is doing, then, we don't control for things like when did our subjects last eat? How much sleep did they get? Have they had caffeine during the day? Most of the time, we don't control for the time of day, for where a person is in their circadian rhythm and so on. And for the kinds of studies that we run with reasonably small samples, this can appear as random noise, when in fact, even if it's not the core thing that we're interested in, the core phenomenon that we're interested in, we could be measuring some of these things and absorbing some of that variance to give us more precise estimates of our effect sizes, which could actually help with the current quandary around replication around the current quandary with replication. And just to give you one example, these are the preliminary results from an ultra-high field brain imaging study, a 7 Tesla brain imaging study where we have people performing a cognitive control task, an end-back task. This is the contrast where we're comparing 3 back versus 1 back, right? So subjects have to say whether or not the stimulus on a given trial is the same as 3 trials ago versus 1 trial ago. And what you can see, the orange blobs are here are activity, and you can see that there are various regions of the salience network which are showing an increase in activity here. But you also see something that you don't see on 3 Tesla scans, which is the increased activity in the periaqueductal gray, which is a key region that regulates your autonomic nervous system, among other systems. So the point is that the evidence is there. We just have to design our experiments and develop our technology sufficiently so that we can see it. And so the hypothesis here is that the default mode network, what it's doing is really using past experiences to construct the beginnings of predictions as ad hoc concepts. Right? So in the past, this network has been understood to be, or interpreted as, creating mental models, creating reality, creating mental time travel, being an experience simulator, being a construction network. And so what I'm suggesting is, consistent with all of these hypotheses, but I'm going a step further to suggest that the default mode network is actually crucial to the ability to construct these abstract ad hoc concepts. And just by way of a little bit of a teaser, you can see that brain imaging studies of concepts and motion concepts actually do show robust activity in the default mode network. And I'm just here, displaying the various, when I'm speaking to audiences who are not experts in neuroscience, I usually walk through this much more systematically. The second hypothesis here is that the salience network, which in the past has been described as an affective network or important for evaluation or for cognitive control or as a multi-use network when subjects are engaging in a task, that really what this network is doing is using the past to predict which prediction errors are worth encoding. You can't encode all prediction errors, right? That is metabolically costly, and your brain is metabolically frugal, at least if you have a normatively functioning brain. And so really you can think about what the salience network is doing is making predictions about what is likely to be important to, what do you have to learn, what do you have to invest your metabolic energy in to learn so that you can predict better the next time. This is basically helping in predictive coding speak, we would say this is helping to set the precision estimates, the reliability of incoming prediction errors so that you know what to learn. And this helps us understand, for example, why we saw the salience network show an increase in activity in over 5,000 imaging studies. And the answer is, if you have a continuously predicting brain where a prediction in one instance is contingent on the sensory array in the prior instance, and you stick that brain in an experiment where you have independent trials that have been randomized to have as little to do with each other as possible, you are forcing the brain into a mode of prioritizing prediction error. So basically, we do experiments with independent trials to make it easier for us to analyze data. But that's making it hard for the brain to predict from moment to moment, trial to trial. That's what you have to do when you want to make your trials independent. But that is a fundamental, that's discontinuity with actually how the brain works. As a consequence, we see the salience network all over the place. And in fact, there is also evidence that these two networks regulate attention, right? Not just, mostly the salience network, although we do see one or two default mode, we also see a couple of default mode regions in here as well. So here what I'm showing you are brain regions that connect to nuclei on the brainstem that originate the brain's neuromodulators. Norepinephrine in the locus coeruleus, serotonin in the raphe nuclei, and dopamine in the substantia nigra and parts of the VAT, and parts of the ventral tegmental area. So we can see that, and again, these blobs, are not activity, they're connections. And so we can see that the brain regions that regulate the body, that maintain allostasis, that create concepts, also control the nuclei that originate the brain's neuromodulators that control the brain's attentional matrix. And a recent paper from Danny Bassett's group at Penn used something called network control theory to provide computational evidence that supports these hypotheses. So her group found that the default mode network efficiently drives the brains into states with little need for energy input, right? Very efficient. That is, we can think of that as prediction. And the salience network, along with the frontoparietal control network, drive the brain into states that require more energy, which we can think of as the encoding of prediction error. And the hubs synchronize neural integration across the brain, which replicates other studies by Van de Kugel and Sporns and so on. And so we can think about this as the architecture of meaning making. So the working hypothesis is that predictions are concepts, that a completed prediction is the categorization of sensory inputs for the purposes of action, and that these categorizations basically not only guide action, but as a consequence, they also construct your experience. And so when we're talking about emotion, we can say, when the brain is constructing an emotion concept and using that concept to categorize sensory inputs and guide action, the brain is constructing an emotional event. Now, in a whole other talk, we could discuss where does the brain get these emotion concepts from in the first place? What is the role of development and culture? And all of those things are really important things to discuss, but not things that you can discuss in a single hour. So what I will do now is just wrap up and say, your emotions are not reactions to the world with essences. They are your constructions of the world. Or more precisely, they're your brain's understanding of what is going on inside your own body in relation to what is happening in the world. Categorizing what's going on in your own body in relation to what's happening in the world. Emotions that seem to happen to you are actually made by you in a highly situated way. So when you feel suddenly, overwhelmingly angry in a meeting, there is not this didn't happen because some circuit fired in your brain. It's because your whole brain predicted, based on your past experience and the current situation, whether or not your heart should race, whether or not you should scowl or smile, whether or not you should sit quietly and plot the demise of your enemy, or you should, slam your fist on the table, and predicting that anger is the cause of these changes. So this helps explain why we have the experience that emotions cause our actions, when in fact, really what's happening is that we're using past experiences of emotion to create ad hoc concepts that guide action and experience construction in the present. A fuller discussion of these ideas and the science behind them can be found in my book. So this is my obligatory, self-indulgent slide here, as well as the scientific papers from my lab. And I should also thank my lab, who really actually did all the hard work that I showed you today. I would be nowhere without these folks, as well as the 50 or so undergraduates who work in our lab every semester, who actually run the studies. And of course, thank the funding agencies, and thank you very much for your attention today. Feel free to give me a shout if you have questions. Thank you.